morning and really welcome to Mayo Clinic for the third annual Impact Symposium. I'm Katie Campbell and I've directed this program since we launched it in 2014. So though the idea of the Impact program was really conceived at Mayo Clinic, its energy and continued success is really a result of all of your hard work. So today's symposium is a celebration of all of you. This year, we really had an outstanding turnout of students and faculty from across Minnesota. Here today, we have impact teams from 20 different colleges and universities across the state. And I'd like to thank everybody for making it here today, especially those of you who've traveled from great distances. So I'm really looking forward to meeting all of you throughout the day and hearing about your exciting hypotheses and future aspirations. Each of you should have a folder with the schedule of today's events. I'd like to briefly highlight some of the key events of today. The day is broken into 3 sessions, and students from each impact topic will either be presenting oral presentations, poster presentations, or lab tours during each of the sections of the day. We'll all reconvene, all of the impact students will reconvene here in Geffen Auditorium for a lunch with a panel of students from the Mayo Clinic College of Medicine, including Mayo graduate students, a Mayo medical student, and a student from the School of Health Sciences. During this lunch session, faculty, sponsors, and any faculty in attendance are invited to participate in an open dialogue with the Dean of the Mayo Graduate School. That's going to take place upstairs on Ganda 5, 104. So if you take the staff elevators to the 5th floor, you can follow signs for Leighton Lecture Hall. And then at the conclusion of today's symposium, we'll be taking a large group photo on that grand staircase that you all came down on the way to the posters. That's at 5:30, and immediately following that photo, there's a closing reception for all participants, faculty, and symposium volunteers. And we hope that this provides an additional opportunity for you guys to connect with impact students from other schools, as well as the students, researchers, and clinicians here at Mayo Clinic. So overall, today's symposium is really an opportunity for us to connect as colleagues working toward a common goal. The Impact Challenge questions for this year's program are real questions that we are struggling with as researchers and physicians here at Mayo Clinic every day. We truly value your innovative ideas and look forward to discussing with you your hypotheses. And now it's my pleasure to introduce Taylor Wells, one of our last year's Impact winning team students, to talk about how the Impact program has really changed her post-graduation career path. So thank you all, and I'm really looking forward to today.
Okay, so first of all, congratulations. No matter which team walks away from today's symposium and from the Impact program with medals, you've all accomplished something really impressive. Developing a novel hypothesis for a real-world health problem is no small feat, and something you should take a great deal of pride in. So as Katie mentioned, my name's Taylor, and I was a 2015 Impact participant. I also want to introduce Andrew Yang. He's back there with the camera. He was one of my teammates last year. So he's a computer science major at Carleton, and I'm super glad he could make it today. So as part of winning the gold medal for our proposal on the underlying cause of hypoplastic left heart syndrome, Andrew and another member of our team, Charles Ironman, conducted research in Dr. Tim Nelson's lab over the summer here at Mayo Clinic. It was a fantastic experience, and we're actually still continuing some research on it right now with the hope of publishing a manuscript on our summer work. So just a little bit about me. I graduated from Carleton College last spring with a degree in biology, and after my summer internship, I transitioned into a position as a research assistant in Dr. Nelson's lab in the Tannen-Kieran-Wannick Family Program for Hypoplastic Left Heart Syndrome. So I started out college thinking I wanted to make nature documentaries, and then it was study lion behavior as a zoologist, and then it finally hit me that I want to be a physician. To say I was a little behind was definitely the truth. It was not only that I had to take pre-med coursework, but I also had to gain a better understanding of medicine and medical research. The Impact program provided me with exposure to medical research that I know I couldn't have gotten from my coursework alone. And as you all know so well, the Impact program really pushes you far beyond that of other undergraduate research programs in asking you to both design your own hypothesis and project. That's an amazing opportunity, and I'm really happy to be here today to support this outstanding program. Think of that first day, that first week, and even that first month of thinking of your impact proposal and working on your proposal. Those were tough times. I remember my first task was just figuring out what HLHS stood for. Yet somewhere along the way during those hours of research that sucked away the precious hours of sleep you get in undergrad, you underwent a crucial transition. A transition from merely taking notes, reading, highlighting, things you do all the time for your classes, to synthesizing, to developing novel ideas, and to creating something that's truly your own. You may not realize it now, but that's a really amazing skill to have, because however you choose to use your talents in the future, your ability to generate innovative ideas will really set you apart. Andrew and Charles and I can attest to the fact that we went through a similar transition. We started off slow for sure, but somewhere along the line, our own transition occurred, and in the nick of time, our hypotheses came together. This is where you guys are all at today. Your minds are full of ideas, excitement, and nervousness. Please be engaged, please be excited, but please don't be nervous. No one presenting a poster or an oral presentation here today has created a perfect proposal. Even though our team won the gold medal last year, we were definitely nervous. I can't speak for my team, but I do know that as I stood up here presenting our hypothesis, I couldn't stop thinking of all of the things we hadn't considered or the possible questions we might get that we didn't know the answer to. But again, please don't be nervous. Be aware of your errors, but don't dwell on them. Stay focused on what you know and present with confidence. Take it all in. There are amazing projects here today. Really engage with the other groups. Ask them questions, give praise, and think of ways that your projects both oppose and build upon others. So that's my take-home message for today, is take it all in. Throughout the day, you'll have the opportunity to connect with physicians, research scientists, and grad students. Take those opportunities, ask them questions, bounce your ideas back and forth, and show them your excitement. Later on in the day, you'll also have an opportunity to tour lab spaces here at Mayo. And I hope you're looking forward to that, because it will not only give you a first-hand look at the research labs here at Mayo, but it will also give you time with a small group to ask questions and to learn about the amazing research that takes place here every day. Today is really special, and I can promise you that in a few years, when you've forgotten the details of your proposal, you won't forget today, so please enjoy it. So I thought the impact program would be a great way for me to start transitioning from science and labs of college to the real world. But I would never have guessed the way that it really would impact my life and affect me. Your ideas can make a difference, and the Mayo Clinic does take them very seriously. Before the impact program, I would never have pictured myself in meetings with brilliant doctors, grad students, research scientists, and research techs. I didn't think I was prepared for something like that right after graduation. I didn't feel like I was really ready for anything outside of coursework. But I was wrong. Now I wake up every day excited to come to work and with a mindset that I am capable of contributing to the field. That's the sense of empowerment that the impact program gave me, and regardless of where you guys end up and where your educational and professional paths take you, I hope the impact program has done something similar for you. So best of luck today, and please say hi and ask me questions throughout the day. So thanks, guys.
Mine, but I thought maybe one of those was yours.
She'll be around all day, and I really hope you get a chance to meet her and all of the other volunteers who are helping make today possible from different research labs across the HLHS, bipolar, and ovarian cancer groups. Now I'd like to turn the mic over to Dr. Scott Kaufman, who's the chair of the Department of Molecular Pharmacology and Experimental Therapeutics, which is the department sponsoring all of this year's impact questions, to really kind of introduce this year's scientific challenges.
Well, thanks, Katie. Welcome to all of our guests. Just a word of Taylor, welcome to the world of science. Whenever I go to give a seminar, I never know what the questions are going to be and whether the audience knows more about the topic than I do either. So I'm nervous when I get up to speak. What I'd like to do is talk for a couple of minutes about molecular pharmacology and who we are, and welcome you on behalf of not only molecular pharmacology, but also Mayo Graduate School. So Mayo Graduate School has 7 areas of specialization where you can get a PhD. Some of those are probably familiar to you, such as biochemistry, physiology, immunology, and some are less familiar, like biomedical engineering, virology and gene therapy, and pharmacology. So who are we? Who are pharmacologists? I'll tell you who we're not. We're not pharmacists dispensing medications. We're not pharmaceutical scientists figuring out how to formulate medications so that your body can absorb them and metabolize them properly. And we're not medicinal chemists, by and large, making new molecules. What we are is a group of scientists who are interested in coming up with new therapies for a wide range of diseases by going into the laboratory, studying pathways that either are altered or are lost in various diseases, and trying to figure out how we can impact those pathways to come up with new treatments. In a sense, I think our Dean for Research has it right. He says the reason we have research at Mayo is to find solutions for patients who don't have solutions right now, and I really think that's what molecular pharmacology is about in a major way. So let me give you a few examples of who we are and what we do and how that led to the impact questions for today. So in pharmacology, we really have three major themes. One is cardiovascular pharmacology, and the sorts of questions that our cardiovascular pharmacologists ask are things like this. Mike Ackerman asks, what causes sudden death, particularly in teenagers and athletes? And if it's due to conduction abnormalities in the heart, what medications can we come up with, and how can we tailor those medications to the specific conduction abnormality that a young person has? Tim Olson asks, what are the causes of congenital cardiac conditions, and how can we improve them? Naveen Pereira is asking, can we use the tools of precision medicine to improve therapy of cardiac drugs? And we're talking about things like antihypertensives, lipid-lowering agents, and drugs that help the heart to pump better. And Katie, Tim Nelson, Andre Terzik, Ada Bafar are asking, can we use the principles and tools of regenerative medicine to repair the heart in those who have either a heart attack, and so the heart muscle is damaged, or in those who have congenital heart conditions? And so their impact question that they brought forward this year is, what's the underlying cause of hypoplastic left heart syndrome? So that's one area of pharmacology. Another area is neuropharmacology or psychopharmacology. And so Eugenia Trishina is asking, can we develop drugs that prevent neurodegeneration, for example, Alzheimer's disease? Suzanne Tai is asking, what are the causes of antidepressant failure? Why do 50% of patients put on antidepressants fail to respond? Can we predict it, and can we overcome it? Steve Rimajoin is asking, can we develop gene therapy to treat cocaine addiction? Dusip Choi is asking, can we develop individualized therapy for alcohol addiction and the coexisting disorders that come with that? And so this group brought forward an impact question of what's the non-genetic cause of bipolar disorder. And then finally, the cancer group. So we have a fairly large cancer center. 30 to 40% of all patients we see at Mayo Clinic have cancer, and this group is actively involved in trying to find new cancer therapies. And so some of the things that they ask are, how do chemotherapy drugs kill cancer cells? And so this diagram up here is actually a wiring diagram for cell death pathways in cancer cells. And the groups who work on anti-cancer drugs are asking what's going wrong in cancer cells that they sometimes don't respond to anti-cancer therapy. Others in the group are asking what's going wrong with DNA repair in cancer, and can we exploit those DNA repair defects as a means for treating cancer? Another group, a couple of investigators are interested in what's going wrong in cellular metabolism in cancer. Can we exploit those metabolic changes to treat cancer? And so this group brought forward, there's another series of laboratories that are interested in, again, in precision medicine, in individualizing cancer therapy. And so this group brought forward this question, which is, what's the cause of sporadic fallopian tube epithelium transformation that leads to ovarian cancer? Now, why ovarian cancer? Because ovarian cancer is the single most lethal cancer in women. has a 70% mortality rate once it's diagnosed. And women who develop ovarian cancer have an average survival of just less than 3 years. Okay, so at a place like Mayo, Taylor and Katie referred to the fact that, that we have medical research going on all around us. And so as part of an ovarian cancer group, I actually find myself surrounded by a whole team of investigators who are interested in ovarian cancer, and they're taking a wide range of approaches. And I think it illustrates the breadth of research at an institution like Mayo Clinic. So in addressing the question of how to better diagnose and treat ovarian cancer, we have a group that's interested in the molecular epidemiology. What are the risk factors, and can you identify them at particularly high risk? We have a group interested in the genomics. What's gone wrong with the genes and the epigenome in ovarian cancer? And a group of investigators interested in the cancer biology. Why does ovarian cancer stay within the peritoneal cavity when other tumors often metastasize widely? But when it stays within the peritoneal cavity, why is it that ovarian cancer often sits down on the bowel and causes bowel obstruction? There are groups interested in various therapies, so endocrine therapy for ovarian cancer, immunotherapy for ovarian cancer, gene therapy for ovarian cancer, and chemotherapy for ovarian cancer. And so what I want to illustrate is that it really takes a big community to address even one cancer. And so Mayo is a place where, in fact, there are multidisciplinary teams around hypoplastic left heart syndrome, multidisciplinary teams around each cancer. And so you can see the types of individuals that get around the table once a month when our ovarian cancer specialized program of research excellence meets. And it's really a village. Folks who are interested in genomics, tumor biology, surgeons, medical oncologists, immunologists, biostatisticians, bioinformaticists. You pick your discipline, there's a way to contribute. We have about 30 principal investigators just looking at this one cancer alone, and they self-assemble in working groups around various projects. So again, what I want to end with is welcome. We're really glad you're here. We hope you have a wonderful day. Modern medical science is really team science. No matter what discipline you're interested in, you can contribute. And you can start contributing now through your ideas. And we welcome your presentations, your thoughts, even as we tell you a little bit about Mayo Clinic. So with that, what I want to do is I want to thank Katie Campbell for organizing this, because if it weren't for her hard work, this entire day wouldn't exist.
Thank you, Dr. Kaufman. And we're lucky to have Dr. Kaufman here with us all day today, so hopefully you guys will get a chance to introduce yourselves and meet all of our faculty here. So as we wrap up this introduction session, I want to kind of give the lay of the land of where everybody's going next. So all of the people who have worked on the HLHS topic, you guys will move right back out into Landau Atrium to start your poster presentation until 12:30. The bipolar disorder groups will meet their escorts in the back of Geffen Auditorium. They should have numbers that correspond with your lab tour number. So find each other, and then you'll proceed to the Guggenheim building for your lab tours. And then the ovarian cancer groups stay put here in Geffen. We're going to start right away with your oral presentations at 10:30. With that, I think we can break for session A, so enjoy.
Well, again, welcome to Mayo Clinic. Welcome to this session, which is going to be focused on ovarian cancer. As we get started, I want to introduce a few of my colleagues, and these are some of my fellow members of the Ovarian Cancer Research Program here at Mayo. First one would be Larry Karnitz, who's the chair of oncology research, professor of pharmacology, and works on ovarian cancer in his laboratory. Kim Calley is a PhD immunologist who's the coordinator for the Ovarian Cancer Research Program. John Varoha is an assistant professor of oncology interested in developing better models for ovarian cancer. I just completed the paperwork to put John up for assistant professor of pharmacology as well. It's submitted, and I'm hoping. And I'm trying to see, because I saw another of our investigators here a short while ago, but I don't see her now. Vici Sridhar? She'll probably be at the poster session. Okay. So again, welcome to all of you. We have the following impact question, which is what's the cause of ovarian cancer, essentially, in women who don't have a genetic predisposition? Just a few words about ovarian cancer, and then we'll get to the presentations. This is the deadliest gynecological malignancy with a 70% case fatality rate. Lifetime risk is 1.4% in the general population, but up to 50% for women with BRCA1 mutations, and substantially elevated for women with other mutations. And we're really still learning the risk alleles for this disease because it is rare enough. It's often diagnosed late because of limited symptoms and very nonspecific symptoms. And high-grade ovarian cancer, the most common and most lethal type, is thought to develop in the fallopian tubes, although there's still controversy back and forth as to how much develops in the fallopian tubes and in how many cases it develops in the ovary. So with that as a background, the only other thing I would tell you is the treatment options are limited. All of us are working on improved treatment because the women we see, by and large, are women who've already developed ovarian cancer. But if we could understand how ovarian cancer develops better, we would have better options for trying to prevent it, particularly in women who are at high risk because of positive family histories and known or unknown mutations that are causative. And so that's really what today's symposium is about. We had 16 proposals. We couldn't accommodate all 16 of you to present, so we picked 8 of the proposals to present. And so the first one, I'm challenged when it comes to computers, but maybe I can get rid of my presentation and get us back up here. First presentation would be this one here from St. Olaf. And so if the team could come up here, I'd love to have you present.
Hi, I'm Zachary Ray-Johnson.
I'm Charlotte Elwell.
I'm Caitlin Van Lith.
And I'm Zachariah Fitz.
We'll be presenting our hypothesis on the sporadic transformation of fallopian tube epithelium to high-grade serous ovarian carcinoma. We believe that the repeated inflammation caused by ovulation leads to a transcriptionally associated mutation in the TP53 gene as the first step on the pathway to tumorigenesis.
We will walk step by step through our rationale and our ultimate hypothesis, beginning with why we chose to focus our research on high-grade serous carcinoma. While there are many types of ovarian cancer, high-grade serous carcinoma accounts for the vast majority of diagnoses and is also generally diagnosed at a later stage than the other types. This late diagnosis and the severity of the condition ultimately leads to a dramatically lower 5-year survival rate for high-grade serous carcinomas than other ovarian cancers. Additionally, 90% of HGSCs are sporadic in origin, and there's a consistent biological marker, as 96% of cases exhibit a TP53 mutation. It suggests a common pathway to tumorigenesis. Because of this common marker and the gravity of this condition, we decided to focus our research on HGSCs.
As mentioned, 96% of HGSCs contain this TP53 mutation. TP53 is incredibly important to cellular functioning. It is normally expressed in response to DNA damage and oxidative stress, and its job is to prevent the cell from developing into a tumor, thus its classification as a tumor suppressor gene. When the cell experiences the DNA damage and oxidative stress, the p53 protein either stops the cell cycle in order to give the cell time to repair the damaged DNA, or it triggers apoptosis within the cell. Both of these mechanisms lead to the cellular and genetic stability of the surrounding tissue. However, when TP53 becomes mutated, as it so often does in high-grade serous carcinomas, the cell is no longer able to perform these regulatory functions. If TP53 is mutated and additional DNA damage occurs, the cell can no longer produce a functional p53 protein to stop the cell cycle to repair the damaged DNA or trigger apoptosis within the cell. Instead, the cell continues to grow and divide in its damaged and mutated state, setting the stage for additional mutations and eventual development of tumors.
While these tumors are found on the ovaries, general scientific consensus now holds that HGSCs originate on the distal fallopian tubes by the fallopian fimbriae. Gene expression studies have confirmed that HGSCs have more in common with fallopian tube epithelium than with ovarian surface epithelium. This, along with several other pieces of supporting evidence, strongly suggests that HGSCs originate on the fallopian tubes and migrate to the ovarian surface. While HGSCs might not begin on the ovaries, the ovulatory hormonal cycle does play a role in setting the stage for high-grade serous carcinomas. A spike in two hormones, estrogen and luteinizing hormone, trigger ovulation. When ovulation begins, the ovarian follicle ruptures, releasing an ovum and follicular fluid. This is an actual physical rip in the ovary that needs to be repaired, and it causes an inflammatory response.
In that roughly 5 milliliters of follicular fluid that comes out along with the ovum, there are also a wide variety of leukocytes. These leukocytes are recruited to the site to repair the damage to the torn ovarian epithelium. While they're digesting damaged ovarian epithelium cells, they release a high number of reactive oxygen species, known as ROS. While these ROS do serve an important function in signaling between leukocytes and they coordinate cellular repair, they also pose a risk to nearby cells such as the fallopian tube epithelium. Exacerbating this problem, leukocytes downregulate the production of superoxide dismutase, SOD2, which is a critical protein in the breakdown of our active oxygen species. Without this protein, the ROS are allowed to persist for much longer than usual, creating a highly oxidative environment. This, combined with this combination, produces a repeated inflammatory environment that can cause a transcriptional mutation in the fallopian epithelium T53 gene. This type of inflammation has been implicated in other types of cancers, such as colorectal cancer, as a leading cause of tumorigenesis. Lending additional support to this hypothesis that ovulation leads to the development of high-grade serous carcinomas, there is a statistically significant correlation between women who experience fewer ovulatory events in their lifetime through either taking birth control, having multiple pregnancies, or experiencing early menopause, and a decrease in their prevalence of ovarian cancer, suggesting a strong link between ovulation and the onset of tumorigenesis.
We believe that this link between ovulation and HGSC is the estrogen-regulated protein interleukin-1 beta. In a study that exposed human cells to interleukin-1 beta, they saw a dramatic upregulation of the TP53 gene. A different study, which exposed bovine FTE to human follicular fluid, saw a dramatic decrease in the amount of SOD2 produced. This is interesting because you would expect the high concentration of ROS of human follicular fluid to upregulate SOD2 because SOD2 is such an important part of breaking down the reactive oxygen species into less harmful compounds. However, because those ROS are so important in the signaling between the leukocytes, SOD2 is downregulated in order to keep that pathway open, and it creates a highly oxidative environment. So that means that this upregulated and unwound TP53 gene is exposed to high concentrations of ROS. Studies in yeast have shown that more transcriptionally active and thus unwound genes are more likely to mutate than those in a condensed chromatin state. What we expect is that the upregulated TP53 undergoes such a mutation. This leads to an unregulated cell cycle and the ultimate development of HGSC.
To summarize our hypothesis, we believe that the inflammation associated with ovulation is the ultimate cause of HGSC development. Inflammation has previously been linked to other forms of cancerous growths. The leukocytes recruited to the ovaries during ovulation produce cytokines like interleukin-1 beta and signaling molecules like reactive oxygen species. The lower levels of SOD2 in the fallopian tube epithelium would result in higher levels of reactive oxygen species, since SOD2 is essential to breaking those species down. These reactive oxygen species are known to cause DNA damage. TP53 is also upregulated and unwound in response to this inflammatory environment, and it's known that more transcriptionally active genes have higher rates of mutation. Taken together, we believe that the unwound TP53 gene and the higher levels of reactive oxygen species set the stage for the mutation of the TP53 gene and the onset of high-grade serous carcinoma. To test our hypothesis, we would want to use cultures of fallopian tube epithelium, but there's a rather limited supply of this sort of tissue, so we could use a method published earlier this year to differentiate induced pluripotent stem cells into fallopian tube epithelium. We could then use these cultures and expose them to increasing levels of human follicular fluid to observe the effect on the rate of TP53 mutation. We expect that at higher concentrations of human follicular fluid and higher levels of interleukin-1 beta, that we would see increased rates of TP53 mutation.
This discovery would be highly significant. If the periovulatory environment really does promote the mutation of TP53, steps could be taken in order to minimize the detrimental effects of ovulation. For example, the number of ovulatory events a woman experiences in her lifetime is highly controllable. From oral contraceptives to IUDs, many women already take control of this event. Additionally, research could be done to explore the inhibition of inflammatory proteins in the follicular fluid. Perhaps these proteins can be inhibited at an optimal level as to not affect their normal functioning while simultaneously stalling the onset of the TP53 mutation. In fact, one study found that there is a statistically significant correlation between a daily dose of aspirin, which is an anti-inflammatory agent, and lower rates of ovarian cancer in women. These discoveries, along with their potential preventative measures, have the opportunity to significantly lower the rate of ovarian cancer in society.
With that, we would like to thank the Mayo Clinic Impact Competition, our faculty advisor, Dr. Kim Campbell, and St. Olaf College for offering us help and support throughout our project. Thanks for your attention. We're now open for questions.
We have time for a couple of questions.
Yes.
So it's my understanding that to some extent that highly proliferating or out of control cells will stimulate an immune response. And since a lot of women are fully ovulating and they're not all getting ovarian cancer, what do you propose is allowing these TP53 mutated cells that might normally be picked up by the immune system to survive in your model?
The functional TP53 protein is one of those essential components in the immune response. It's how the cell gets tagged by leukocytes for destruction in immune response. So without a functional TP53 protein, you can't have that kind of response.
Go ahead.
So I know that P53 is one of the most common mutations in inherited ovarian cancer, right? So it's present in the germline. But have there been studies that have shown that it's also equally common in sporadic causes of ovarian cancer?
Can you repeat the question?
So P53 is common in inherited ovarian cancer, right? So it's present in the germline. But what we're trying to address here is the sporadic cause of ovarian cancer. So is P53 equally common? P53 mutation equally common?
It is. The vast majority of high-grade serous carcinomas are sporadic in origin, and the vast majority of those have a TP53 mutation.
Right, so it's 90% of all high-grade serous carcinomas are sporadic. and 96% of all high-grade serous carcinomas have a TP53 mutation.
Dr. Karnitz, you had a question?
Yeah, so the key point in your hypothesis is that TP53 gene is unwound, and that makes it more sensitive to oxidative damage. In your experiments that you proposed, I didn't see how you were going to distinguish between the unwound version being more sensitive versus a gene that's not unwound.
We know that exposure to high levels of interleukin-1 beta unwinds TP53, so we could control the amounts of interleukin-1 beta in one control scenario, and we could control the amount of ROS as two independent variables and run multiple trials that way.
Last question, Cordelia.
How do you propose to track the mutation rate of TP53 in your control and your treated plates?
We know that TP53 is found on chromosome 17, so even if it's a little bit challenging, you can sequence chromosome 17, and you also have clear signs of tumorigenesis, which you can observe for, in which the nucleus becomes, you can see change in the nucleus becomes darker, and the cells lose their contact inhibition and start to pile up in the petri dish rather than spreading out in a single layer.
But I think where Cordelia was going is that you're probably going to have to do single-cell DNA sequencing to see the P53 on a cell-by-cell basis in that experiment.
Right.
Yeah.
Okay. So thank you very much. So while I change the slides and ask group 2 up here, I'd like to introduce a couple more folks who are in the audience. Here we go. Have Hu Lee, who is a member of the ovarian cancer group. You'll meet him this afternoon. He's a systems biologist, systems pharmacologist. Cordelia McGehee, who is an MD PhD student. She'll be helping with some of the tours. And Dr. Kazezi, who is an immunologist interested in ovarian cancer. So welcome to all of you. Come on up.
Good morning. I would first like to start off, by introducing ourselves. My name is Nathan Peterson.
I'm David Osmond. And I'm David Campbell.
Before we address the question regarding the cause of sporadic fallopian tube transformation to ovarian cancer, I would first like to draw your attention to some background information as a way of explaining why this topic is so important. Ovarian cancer remains among one of the most lethal gynecological malignancies for women, with a 5-year survival rate of only 45% due to a lack of early screening and the fact that this cancer often isn't diagnosed until it is detected in advanced stages. Sharing from a personal story, my grandmother on my mom's side died of ovarian cancer just 6 months after she was initially diagnosed in 2012. Her rapid decline was difficult to see, and there was no known prior history of ovarian cancer in my family.
Similarly to the story Nathan shared, in 1991, unfortunately, my grandmother also passed away from ovarian cancer complications. And from what my family could recall, there was no history of ovarian cancer. Within the general population, the lifetime probability of developing ovarian cancer is 1.4%, as Dr. Kaufman pointed out. It was estimated that 225,500 women each year worldwide are afflicted by the disease, and that ovarian cancer claims the lives of around 140,200 each year.
So, high-grade serous carcinoma is among the lethal types of ovarian cancers. It's become evident in recent years that HGSC originates in the distal fallopian tube epithelium as serous tubal intraepithelial carcinoma. The actual site of origin, as well as the pathogenic process, is still a topic of much research. So for our research, we propose a link between tissue damage caused by incessant ovulation, hyperactivation of nuclear factor kappa B, also called NF-kappa B, or REL, and sporadic transformation of the distal fallopian tube epithelium into ovarian cancer.
One long-standing hypothesis is the incessant ovulation hypothesis, which proposes that ovarian cancer arises from epithelial tissue and DNA damage caused by a cyclical release of ovum from the ovaries without disruptions due to factors such as pregnancy or birth control. It's been observed that incessant ovulation can cause localized tissue damage, and that cortical inclusion cyst formation can lead to dysplasia and in turn ovarian cancer. Furthermore, the distal fallopian tube epithelium in particular is noted to undergo continual damage and regeneration during the reproductive years, which could lead to tumor formation and suggest a tubal origin for ovarian cancer. Another established theory is the gonadotropin theory, which focuses on the role of reproductive hormones and particularly on the increasing levels of localized estrogen in relation to ovarian cancer. Although both of these theories are conceivable, they discount a common mechanism. Nuclear factor kappa B, or NFKB or REL, is a transcription factor involved in a variety of cellular responses, but is specifically known for its role in promoting inflammation. NF-kappa B is typically latent and inactive in the cytoplasm due to a family of inhibitor of NF-kappa B or I-kappa B proteins. However, inducers such as extracellular stress and DNA damage can lead to phosphorylation from the I-kappa B kinase or IKK complex, resulting in degradation of I-kappa Bs via ubiquitination and subsequent activation of NF-kappa B. Once activated, NF-kappa B can enter the nucleus and perform its regulatory roles in gene expression.
Because of this link between inflammation and cancer, NF-kappa B itself has been a topic of study in relation to tumor development and cancer progression. In fact, NF-kappa B has been linked to a number of epithelial cancers, including ovarian cancer. It has been suggested that tissue damage can lead to the hyperactivation of NF-kappa B and that continuous activation of this transcription factor can lead to cancer formation. A critical connection of NF-kappa B to cancer is also demonstrated in its ability to upregulate the expression of tumor-promoting cytokines, inhibit apoptosis, and promoting cell proliferation. Because of this, many current anti-tumor therapies attempt to block NF-kappa B activity as a way to inhibit tumor growth. Furthermore, since NF-kappa B's activity is localized, it would suggest that tumor formation would only occur at the site of tissue damage. Another thing to note is that NF-kappa B has also been cited in its role with promoting the malignancy and migration of transformed cells, which could also be linked to metastasis of STIC to the ovarian surface, leading to HGSC as was described earlier.
So we introduced earlier how incessant ovulation can cause repetitive tissue damage and DNA damage, as well as stress at the site of the distal fallopian tube epithelium, and that in addition to the role of inflammation that NF-kappa B plays in inflammation and tumor progression, hormonal regulation of NF-kappa B has also been observed. Gonadotropin releasing hormone can both activate NF-kappa B and protect cancer cells from apoptosis. In other uterine disorders like endometriosis, lesions are associated to hormonal imbalances, including estrogen synthesis, metabolism, and responsiveness to progesterone resistance. Progesterone, which is also elevated during pregnancy or by the lack of estrogen, may contribute to lower NF-kappa B levels and a reduced risk of ovarian cancer. It has also been demonstrated that 17-beta-estradiol, or E2, interacts with NF-kappa B, and that E2 has an ability to modulate activity of NF-kappa B both via genetic and non-genomic actions.
With all this in mind, we propose the mechanism as such. A continuous release of ovum from the ovaries without disruptions induces damage at the site of the distal fallopian tube. This triggers the hyperactivation of NF-kappa B in the distal fallopian tube epithelium, leading to dysplasia and tumorigenesis. Once STIC is formed, the hyperactivation of NF-kappa B promotes the migration of these cells to the epithelial ovarian surface, leading to HGSC and giving the appearance of an ovarian origin. This hypothesis could be tested using the hen model, whose reproductive system resembles that of the humans. Experimentally induced incessant ovulation in hens has already shown a correlation to ovarian cancer development. This type of experiment could be repeated with an emphasis on monitoring for NF-kappa B activity. Alternatively, stem cells have been found in the fallopian tube and uterus with multipotent and even pluripotent promise and potential. Using these cells in regenerative medicine to create uterine models and artificially inducing NF-kappa B could allow researchers to simulate ovarian cancer and more effectively test for potential therapeutic agents.
So as for preventative measures, it would be important to monitor NF-kappa B activity at the site of distal fallopian tube epithelium in females particularly those who have suffered damage at the site due to a lack of ovulatory breaks. Moreover, finding ways of diminishing or inhibiting NF-kappa B at the site of an injured distal fallopian tube epithelium could prevent cell proliferation and, in turn, prevent tumorigenesis. The inhibition of NF-kappa B activity has been demonstrated with the use of drugs and strategies including penoxidone, curcumin, and Krelox gene targeting. However, because of the positive contributory roles NF-kappa B plays, seeking to discontinue its activity altogether could lead to negative unintended consequences. Instead, by merely decreasing the activity of NF-kappa B at the site of the distal fallopian tube epithelium, tumorigenesis could still be prevented while providing NF-kappa B's beneficial results. Also, in cases where cancer cell has been diagnosed, because apparently active NF-kappa B encourages cell death resistance, diminishing its activity would be crucial to improve the effectiveness of chemotherapeutic drugs and apoptosis and the apoptotic response to radiation therapy, as has been shown.
To remind us why this research is important, as stated earlier, approximately 140,200 women die from ovarian cancer each year. In other words, that's approximately 380 families that are losing their loved ones every day. The more we can discover, the more possibilities there will be for earlier detection and prevention of ovarian cancer for many grandmothers across the world. Thank you.
Time for a couple of questions. Right there.
So our understanding is that the only one common mutation among these sporadic tumors is P53. What is the percentage of patients with activated NF-kappa B mutations, gain of function mutations, or any alterations that increase their levels?
That's a good question.
I don't believe we found any research regarding like the statistics of how much some people had with NF-kappa B versus others, but that would be a good area to look into.
Other questions? So as you were thinking about NF-kappa B inhibitors, which you referred to, do you think it'd be possible to administer them either in an experimental setting or to women who are at risk on a regular basis, or do they have such broad side effects that you'd worry about them?
Yeah, we did find in the research that there are, along with the potential possibility for therapy and prevention, there's also positive effects that NF-kappa B has, so we would probably lean towards favoring using it on artificial or created uterine models or hen models before actually testing it on human. Okay. Yep.
Other questions for this team? Okay, thank you very much.
Thank you.
Hi, I'm Nick Nicholson.
And I am Jacob Stern, and today we would like to talk to you about the estrogen-motivated transformation of tubal epithelial tissue into high-grade serous carcinoma.
So why is this topic important? And as you all know, we all went through the research question, what is the cause of sporadic fallopian tube epithelium transformation to ovarian cancer? And why do we care? Ovarian cancer is the 8th most common cancer within women, and the 5th leading cause of cancer within women. And this is because 75% of ovarian cancer's diagnoses are in advanced stages, which leads to on average, 5-year survival rate of 20%. Within our study, we focused on high-grade serous carcinomas, and this is because 70% of ovarian cancers are diagnosed as high-grade serous carcinomas. And within this cancer, 90% are only diagnosed in the very late stages of metastasis. What defines specifically a high-grade serous carcinoma is its molecular makeup, focusing on the mutation in P53. To this diagram, you see like the tissue type, cross sections of tissue types, and the one we focus on is the left. So anatomically, the fallopian tube and the ovary are connected to the uterus and in very close proximity. They're divided by a thin layer of serous fluid. This is produced by the serous tissue within the pelvic cavity, and is used to reduce mechanical friction between the organs in this area. The zoom-in section of B looks at the fimbriae, the little finger-like parts coming from the fallopian tubes, has two consistent cell types: the excretory cells that release serous fluid into the environment, and then the ciliated cells, which kind of hold on to the serous fluid to each of the organ's tissues. The second one is the ovarian surface epithelium. And what's interesting to note is that this epithelium actually exhibits both mesenchymal and epithelial cell type markers, which we'll go into in just a second. So, most common mechanism of metastasis is epithelial-mesenchymal transition, and this is the transition from an epithelial tissue with mutation into a mesenchymal anchorage-independent cell. And this is defined by 5 separate steps. The first, EMT to detachment, and this is the detachment into either the lymphatic system or the vascular system, but in our case we focus in on the serous fluid. After this happens and it gains a holding within the area, the cell types undergo an EMT transition and attachment. And what we focus on is this transition into the epithelial of the ovary in the ovarian cleft. And once this takes place, tumorigenesis forms and colonization. So, our hypothesis and what we focus in the mechanisms, there are 2 steps. The first part focuses on the initiation of the tumor. And this is driven by estrogen and estrogen metabolite duality. We focus in on how estrogen leads to cell promotion within this area, to normal cells and cancer cells, and how cell metabolites drive the carcinogenesis of the fallopian tube to change into this EMT and MET transition. So finally, the second is the transition from the fallopian tube into the ovary. And this is categorized by the EMT transition into the serous fluid, where it exists for a while, and then gets caught on the ovarian clefts, where it undergoes EMT and starts colonizing within the ovarian serous tissue. So to explain a little bit our hypothesis is what instigates the primary tumor formation. And so here we see the specific chemicals involved within carcinogenesis of fallopian tubes. And these have been proven in breast cancer. And you see the 4-hydroxyestradiol and 16-hydroxyestradiol are derivations of the way body metabolizes estrogen. So then, why is the fallopian tube the specific type of cell that's going to be affected by estrogen? And we work to prove that there's a difference between the estrogen receptor rates, alpha and beta, within the fallopian type cells. Now Jacob's going to explain a little bit more about our hypothesis and into the experiments which we like to try to get to.
So, as Clay mentioned, the first thing I'm going to talk to you about is explain a little bit more about how we propose the cancerous fallopian cells become embedded in the ovarian epithelial tissue. And as Clay mentioned, the first step is the epithelial to mesenchymal transition caused by the carcinogenic estrogen metabolites. These free-floating mesenchymal cells, which are already cancerous, then become lodged in ovarian folds. And ovarian folds are common in older women and post-menopause women, which is the #1 demographic for ovarian cancer. Once stuck in this ovarian fold, the free-floating cell undergoes enough mutations to reincorporate into the ovarian epithelium through a mesenchymal to epithelial transition. And some of you might be wondering, what happens to the primary fallopian tumor? Why is it the ovarian tumors that are killing people? And the answer to this is a difference in growth rates and mutation rates in the ovarian, or in the 2 microenvironments that the tumors are located in. So the ovarian tumor on the ovary is in an environment with a higher level of interleukins, which are growth-promoting molecules, and estrogen, which is also a growth-promoting molecule and induces mutagenesis. Next, I will talk to you about an experiment designed to test the first component of our hypothesis, the role of estrogen and estrogen receptors in ovarian cancer. First, we propose using a Western blot analysis to detect estrogen receptor levels in fallopian epithelial tissue and compare that to ovarian epithelial tissue. Then, we would take fallopian tissue, we would insert a luciferase gene for later bioimaging, and how this works is luciferase is a fluorescent protein that can be observed in vivo in a live organism. And once we've inserted the luciferase gene, we would have 4 treatment groups, or 3 treatment groups and a control. One of these we would silence estrogen receptor alpha, 1 estrogen receptor beta, 1 where we would silence both, and 1 where we would silence none. And we would silence these using molecules called small interfering RNAs, which are molecules that prevent the transcription of mRNAs into proteins. We would take these 4 treatment groups and treat them with carcinogenic estrogen metabolites Clay mentioned earlier, 4-hydroxyestradiol and 16-alpha-hydroxyesterone. We would then inject these into immunodeficient mouse ovaries and observe rates of tumor formation and tumor size via bioimaging with luciferase. This experiment is proposed to test the second component of our hypothesis, the transitional mechanism via the serous fluid. To do this, we would begin by, again, inserting the luciferase gene into fallopian epithelial tissue. We would be growing this tissue in an ex vivo fallopian tube culture system to more closely mimic the in vivo conditions the cells are growing in. We would again induce carcinogenesis with these carcinogenic estrogen metabolites. Then, using a method called FCS, or fluorescent cell sorting, we would identify and isolate any mesenchymal cells that formed, and inject these cells into the immunodeficient mouse serous fluid. And these mice would have induced menopause and be older to increase the likelihood of ovarian folds. We would then observe the sites of tumorigenesis, and we would expect to see tumors forming on the ovary. And what you can see here is a picture of the kind of output you would get from bioimaging via luciferase. So, why is our hypothesis innovative? There's two components: biological innovation, and it affirms that fallopian cells are cancerous before incorporation into the ovarian epithelium. It identifies serous fluid as a mode of transportation for the free-floating mesenchymal cells. It provides a link between estrogen receptors and ovarian cancer that has previously been shown in breast cancer but not conclusively proven in ovarian cancer. And it identifies the importance of ovarian folds. On the technological side, our experiments would utilize three relatively modern techniques, including the introduction of luciferase via gene editing for bioimaging to observe in vivo tumor formation, the use of small interfering RNAs to silence specific proteins, in our case estrogen receptors, and the use of an ex vivo fallopian culture system to more closely mimic real-life environments for growing tissues in a lab. Our hypothesis is significant because it brings a newfound importance on previously understudied or underappreciated therapeutic and research targets, including the reduction of ovarian folds and clefts, the inhibition of estrogen metabolic pathways that lead to carcinogenic metabolites, the engineering of serous fluid to make it less hospitable for free-floating mesenchymal cells, and the use of molecules to inhibit EMT or MET processes. Thank you all for your attention. We would like to thank our advisor, Dr. Laura Burick, for her advice over the past few months, and the Mayo Clinic for this wonderful opportunity. And we'd now like to open things up for questions.
Okay, so we have time for a couple questions right there.
Okay, so I have two kind of separate questions. If you're interested in estrogen's role in tumorigenesis, why is it important to distinguish between ER alpha and ER beta?
Because, as Clay mentioned, we are suspecting that there's a difference between the estrogen receptor alpha and beta ratios in fallopian tissue as opposed to ovarian tissue. And they cause slightly different growth pathways, and we're proposing that this difference is the reason that fallopian tissue is more susceptible to cancer.
And did you look at the intercellular or the nuclear receptor of ER beta and look into any like signaling pathways that contribute to tumorigenesis and other growth factors?
We didn't look into the specifics of the growth pathways, but we know that they do promote growth.
Okay. So you wouldn't at all propose like using like an aromatase inhibitor to like stop estrogen's role in general in tumorigenesis, or an estrogen-like antagonist at all in your experimental design?
We could, but I think the use of siRNAs would do pretty much the same.
Yeah, I really like your idea of EMT motility and preferential survival in the ovary environment. But how do you reconcile your hypothesis of estrogen dependence and the fact that ovarian cancer is prevalent post-menopause?
Well, there's three different types of estrogen, and one type of estrogen is upregulated during post-menopause women. And is it estradiol or?
It is estrone, I believe. Actually.
Estrone. Okay, so estradiol is more common in premenopause. Esterone is more common in postmenopause. So it could be that the upregulation in this type of estrogen is enough.
And this would lead to like separate different metabolites that may allow for a different kind of entrance into the cell, causing more DNA damage.
Go ahead.
Yeah. I have a question about the EMT story. So it seems like you propose that the tumor is originated from the fallopian tube and then transferred into the ovary. But I think current standing of metastasis is the tumor have a higher chance of migrating into the EMT when the primary tumor is large enough. So how could you explain that in the high-grade carcinoma, usually the case there is a big primary tumor in the ovary, but there is no primary tumor in the fallopian tube.
So as we proposed, the primary tumor in the fallopian tissue we suspect is overshadowed by the ovarian tumor because of the higher rates of interleukins and estrogen present in the ovarian environment. They cause it to grow faster and mutate faster, so it makes it more dangerous.
So in your reading about estrogens and breast cancer, did you run across the possibility that there are some other pathways that weren't on your slide that will actually give estrogen metabolites that covalently modify DNA? The so-called estrogen catechol, where you put an extra hydroxy on that A ring.
Not that I know of.
Yeah,
There's a really active investigation of that pathway as well that involves estrogen that might link to what you were looking at. Go ahead.
Did you guys find anything, or what's your rationale for the difference between women who develop ovarian cancer and who don't?
Well, because this is the sporadic cause and we discounted any genetic predisposition, we assumed it's just random mutation, and we didn't identify one clear environmental factor that could cause the difference.
Any other questions for this team? Okay, thank you very much.
So we first just like to thank you all for listening to our presentation on the role of folate in fallopian tube epithelial transformation of ovarian cancer. For our primary research, we thought there was a role of folate in fallopian tube epithelial transformation of ovarian cancer. To begin, we'd just like to introduce ourselves.
I'm Emily Facile.
I'm Megan Braun.
I'm Whitney Zenz.
And I am Andrew Studer, and we are students from St. Olaf College. To begin our presentation, we would like to introduce the metabolic role of folate and how the incessant ovulation hypothesis complements our folate hypothesis. Then we will discuss the possible mechanism for fallopian tube cell epithelial transformation and its movement into the ovary. Lastly, we will explain how folate is significant to the risk assessment and study of ovarian cancer, and how it is innovative within the field of regenerative medicine. Just a little bit of background information for everyone in the audience. Folate is a B vitamin, and its normal role in the cell is being a precursor for the adenosylmethionine, or otherwise known as SAM, which is the methyl donor for DNA methylation. So folate, it has metabolism as seen here, is transformed via enzymes into 5-methyl-THF, then transformed to methionine, and SAM, which functions in DNA methylation. Now when you have low dietary folate, that's when some of the problems can occur. Low folate has been known to cause DNA breaks, hypermethylation, chromosomal instability, and some p53 breaks in some studies that were dealing with different types of cell lines. Now, furthermore, a study on colon epithelial cells in mice showed that there was a decrease in cell adhesion and an increase in cell migration with low folate. And we propose that this can be extrapolated to fallopian tube epithelial cells.
We also looked into the incessant ovulation hypothesis, which has been previously well studied. Basically, it says with each excessive ovulation, the ovarian wall becomes weaker and weaker. And so with more, we see this more in older women or women who haven't had any kind of contraceptive. So with each ovulation, we also see a fallopian tube epithelium and follicular fluid entering the ovaries. And this follicular fluid contains free radicals which can mutate and or damage the DNA. And this leads to other harmful effects. Overall, like I said before, with this follicular fluid and with the ovarian wall weakening, older women and postmenopausal women have a higher risk of this cancer.
Our hypothesis states that low folate intake can increase the risk of ovarian cancer by causing p53 hypermethylation patterns, which would lead to uncontrolled cell growth. The uncontrolled cell growth develops cancerous cells in the fallopian tube, which then migrate to the ovary via the fallopian fimbriae, which ultimately leads to ovarian cancer.
So since the question the Mayo Clinic asks us to look at involved the sporadic transformation, we looked into various environmental factors, including dietary supplements that could possibly differ between the individuals who do get this cancer and who don't. One of the metabolites that we found, or one of the vitamins we found that could have an increased risk of of ovarian cancer was found to be folate. Studies were kind of mixed results on whether or not this is actually the case, but the study we found and displayed here was able to find the effect when looking at alcohol consumption in conjunction with folate consumption. So what's happening with the alcohol consumption is when the women in the study, or not in the study, it was a survey and they gave information regarding their dietary intake and other life factors as well that were known ovarian cancer risks. So when the women in the study who consumed greater than 20 grams of alcohol per week and also had lower dietary folate intake, this is increasing folate intake, had higher risks of epithelial ovarian cancer than those who did not. Higher folate was shown to have protective effects against ovarian cancer for those who consumed larger amounts of alcohol. The reason this is happening is because alcohol mimics the effects of low folate because it interferes with folate metabolism, sometimes cleaving folate molecules, and causing more folate excretion from cells.
Now we'd like to turn the attention to what happens to p53 regulation and expression when we have a lack of folate. So in one study we found that in pre-neoplastic cells there was an upregulation and a higher expression of p53 protein. And this was due to the inability of, this hypomethylation was due to the inability of methyltransferases to methylate the DNA. But in another study, we found that in neoplastic tissue, there was a reverse of hypermethylation. With more methylation on the DNA, the machinery wasn't able to read the DNA, make RNA, and transcribe it into proteins. And therefore, with less p53, as we've seen before, it's a cell cycle regulator. Less cell cycle regulation can lead to an overgrowth in cells.
So we continue to look at different types of cancer cells that are different types of cells that are known to have the same p53 mutation because folate and amount of dietary folate intake is not normally, has not been studied so far yet with ovarian cancer. So we looked at the cells of colonic mucosa. So what was found in response to low p53 is that cell adhesion proteins such as proteocadherin 4 and nitrogen and integrin alpha V were all downregulated in some of the mice in this study as a response to low folate intake. And the other thing that was found was p53 was downregulated in older rats with no folate intake as compared to the younger rats who had the same low folate intake, indicating that this would be more of a problem in older women or older female animals in this case. Another study specifically looked at p53 again with just response to folate intake rather than with response to age. This study used a more severe form of folate deprivation in the cells, and it was able to find that when this larger deprivation of metabolic folate was in the cells, that p53 was downregulated with response to the low folate intake.
A way that the cancer can move from the fallopian tube to the ovary is through the fallopian fimbriae. During ovulation, when the oocyte is released, the ovarian wall becomes significantly weaker, and as you can see in part C of the figure, the fimbria can deposit some of its cells into the wall of the ovary. Therefore, if these cells are cancerous, that can lead to the cancer migrating into the ovary from the fallopian tube. As well, the role of folate in ovarian cancer would play an important role in early risk detection. If there's a way to measure the levels of folate in women's blood who are already at risk, that can increase their risk, or our knowledge of their risk as well, which could hopefully lead to detection while the cancer is still in the fallopian tube, rather than by the time it has already migrated into the ovary. Our proposed experiment would be to study the methylation patterns using stem cells of the p53 gene and how what is impacted by different levels of folate.
In addition, we also propose that you can use stem cells as a potential treatment for this, potentially either replacing or being used as a treatment for the already cancerous fallopian epithelial cells. And also, since the incessant ovulation hypothesis states that the ovarian wall has been weakened, we propose that it is possible for stem cells to regain that thickness and to increase the chance of ovarian cancer not forming. So in conclusion, folate has been known to have DNA hypermethylation and some chromosomal instability, which then leads to downregulation of both cell adhesion and p53 regulation. Also, the incessant ovulation hypothesis states that there are a bunch of free radicals from the follicular fluid that can cause DNA mutations, which can cause epithelial cells from the fallopian tube to become cancerous. And then those cells can now migrate to the ovary via the fallopian fimbriae and grow into a tumor that's on the ovary, leading to ovarian cancer. We would like to thank you all for listening to our presentation. We would like to thank the esteemed people here at Mayo for inviting us and all of you for listening. And if you have any questions.
So, does the low level of folate cause the cancerous mutations, or is the hypermethylation of p53 caused by low folate allow for the DNA mutations to occur?
So low folate is able to mimic, or it causes the effect of the first hypo and then hypermethylation of the p53 gene.
Okay. So.
Kim?
So folate is now fortified in all cereals and products as part of a law that changed in 1996 because low folate is known to cause neural tube defects in developing fetuses. And since the folic acid was put, at least into the United States diet, those defects have really gone down. Are you aware of any changes in frequency of ovarian cancer diagnoses in the last 20 years compared to before we started supplementing our diet with folate?
I would think, I am not, but I would think that if folate was involved in causing a risk, that maybe other metabolites in that methyltransferase pathway would also be causing that effect. So if there were a lack of other metabolites in that pathway, that it could also cause the same effect.
As similar to that question, in other countries where they don't have folate incorporated into their diet, so third world countries, things like that, is there a difference in ovarian cancer rates?
That is not something I've looked into.
Okay.
What is the percentage of women in America that have a folate deficiency?
I'm not aware of that number, I'm sorry.
Perhaps another way to think of the same question, because we don't typically measure folate levels in our patients unless we specifically are looking for dietary or issues like malnutrition. One of the consequences of folate deficiency is that patients will develop anemia, and the red cells get to be big, and so we call it macrocytic anemia. And I don't know the answer to this, but do you know the incidence of macrocytic anemia in patients diagnosed with ovarian cancer?
John doesn't know.
No, I'm sorry.
I didn't preface that one. I don't know the answer to that.
And this is somebody who takes care of ovarian cancer patients, and he doesn't know the answer, so.
Yeah. But, as you're looking through, I haven't looked through the folate receptor, the folate literature as you have, so I'm just kind of wondering if you've come across that.
The literature we read for this presentation did not discuss that.
Back there. Yeah.
I think I might totally be off on this, but I thought that there's an understanding that cancer cells in particular really depend on folate in order to continue their proliferation. This is something I don't know a lot about, but I was just wondering, is that an essential nutrient that would be needed for the tumor cells as well? Are they like sequestering the folate that's in the body and taking it for themselves, or how are they able to proliferate if you have this massive folate deficiency?
So we're thinking that the folate, first, it hypomethylates the genome in general because it doesn't have the methyl groups to donate to newly synthesized DNA. So with that, you get the overexpression of some genes that could cause cancers, and then the p53 gene, as was found in these studies, is actually hypermethylated in the secondary response. So it doesn't have those cell cycle checkpoints that would allow the tumor to keep metastasizing.
Other questions for this team? Thank you very much.
Good morning, we are the team from McAllister College, and today we're here to present our proposal on the sporadic fallopian tube transition into ovarian cancer. But before we discuss our hypothesis specifically, we would first like to qualify this with a brief overview of 3 relevant fields. Firstly, we will discuss a little bit more about ovarian cancer and why the fallopian tube epithelium is of particular interest. Secondly, we will qualify this discussion with the importance of inflammatory events, specifically in their ability to precipitate the presence of reactive oxygen species, ROS. And then lastly, we will discuss briefly microRNAs and how perhaps the expression profile of these microRNAs may vary in response to the presence of ROS.
So according to CDC, ovarian cancer causes more deaths than any other cancers in the female reproductive system. And there's a dualistic model that broadly categorizes all of histological types of epithelial ovarian cancer into 2 categories. The low-grade serous cancer is defined by tumors that are still confined inside the ovaries during the early stages, while the type 2 refers to the high-grade serous carcinomas that are defined by tumors that are aggressive in nature and present during the advanced stages. And although controversial, there has been research done that has proposed that the fallopian tube is the site of origin of high-grade serous carcinomas rather than the ovaries.
And as mentioned earlier, part of the reason why ovarian cancer is so lethal is because it's really hard to make an early diagnosis, and by the time physical symptoms such as pelvic pain and bloating arises, the cancer has already spread to either the pelvic area or the abdominal areas. The highly aggressive nature of this cancer suggests an abnormally high activation of the EMT pathway, which further points to the loss of adhesion between cells and the loss of polarity within a cell.
So, as we've seen today, a really new, interesting area of cancer research is in oxidative stress as it relates to inflammation. So I'm just going to talk through briefly an inflammatory response. So in a normal immune response, we have neutrophil activation, and neutrophils usually act as phagocytes, and they also tend to release reactive oxygen species. And now these reactive oxygen species in the immune response are great because they can not only break down invading cells, but they can also, they're also good for cell signaling. And so, good. The costs outweigh the costs are less than the benefits. The benefits being that you're getting rid of the cells, the cost being you're causing cell damage to the host cells. But in cases where you have chronic inflammation, you end up having high concentrations of reactive oxygen species all the time. And this high concentration of reactive oxygen species can in turn cause a lot of molecular damage to the host.
So in order to understand how microRNAs tie into our hypothesis, it's important to discuss what a microRNA is. And these are small non-encoding RNA sequences that are typically 12 to 20 base pairs long. These microRNAs are important in the cell for gene regulation, specifically through transcriptional repression, because they recognize and designate messenger RNAs for degradation. Some of the recent researchers have shown that ZEB1 and ZEB2 are actually inhibited by a specific type of microRNAs. And when you have a dysfunction of this microRNA, these proteins become active, and they in turn inhibit E-cadherin, which is a key epithelial phenotype protein. Another research done in 2011 showed that a different microRNA is important for the cell to mediate unwanted EMT apoptosis, otherwise known as anoikis. And when the cells lose this microRNA, they lose the function to undergo apoptosis when unwanted EMT occurs.
Furthermore, changes in microRNA expression have also been linked to changes in tumor-suppressing transcription factors, which potentially may be another lead-in to a cause for tumorigenesis. However, it is important to note that recent research, especially in 2014 and 2015, has suggested that, in fact, there is quite a large degree of microRNA expression that varies in response to the presence of reactive oxygen species. Therefore, in our hypothesis, we propose that inflammatory events leading to the presence of reactive oxygen species will cause misregulation in specific onco-related microRNAs, which in turn, through their altered expression, can lead through a change from the epithelial phenotype into a cancer cell.
So the rationale of our hypothesis, it kind of links two hot areas of the cause of ovarian cancer together, the oxidative stress and the misregulation of microRNA. And a lot of previous studies have demonstrated that oxidative stress is a key driver of early tumorigenesis in ovarian cancer. Oxidative stress changes the expression of a lot of genes. And thanks in the genes, microRNA is a key regulation mechanism of gene regulation. So it is very likely that oxidative stress could change the microRNA expression pattern and therefore change the gene expression and lead to all of the tumorigenesis abilities. Here we specifically propose 3 microRNAs that may respond to oxidative damage and lead to the cancer cell transformation. The first one is MIR181A. The downstream target of this microRNA is SMAD4, which is an inhibitor of the TGF-beta pathway. It's a tumor suppressor gene. And oxidative stress could lead to the upregulation of this microRNA and therefore lead to the downregulation of SMAD4. And the loss of this tumor suppressor gene would lead to the increased activity of TGF-beta pathway and therefore lead to the fallopian tube transformation into cancer cell. The other microRNA we're proposing are MIR200A and MIR141. Both are in the family of MIR200. And the downstream target of those 2 microRNA are P38 alpha, which is also a tumor suppressor gene. Oxidative stress could lead to the upregulation of those 2 microRNAs and therefore lead to the downregulation of the tumor suppressor T38 alpha, therefore causing normal cell transformation into cancer in the fallopian tube. The third microRNA we are looking into is MIR200C. The downstream target of those 2 microRNA are FN1 and NTRK2. Those 2 genes are crucial for cell migration and anti-apoptosis. And oxidative stress could lead to the downregulation of this microRNA and therefore lead to increase of the expression of those 2 genes and make the normal cells have enhanced cell mobility and anti-apoptosis. Both are hallmarks of a cancer cell. Of course, since oxidative stress causes a lot of changes in the cell, it is also likely that oxidative stress could change the expression changes of microRNA that we didn't mention. And as previous group discussed that TP53 and NK-kappa B, those 2 genes are shown to be dysregulated in the ovarian cancer, and it's also possible that the dysregulation of those 2 genes are due to the microRNA as well. Another part of our hypothesis is it's highly testable in a normal cancer research lab setting. We can use hypoxial cell culture, basically introduce oxidative stress to either primary fallopian tube cells or IPS cells derived from the fallopian tube cells, taking advantage of the regenerative medicine product. And after that, we can test their tumorigenesis ability after oxidative stress. We can test the cell proliferation ability, the colony formation ability in the soft agar, and the tumor formation ability in the nude mice xenograft, which are pretty common tests in cancer research setting. And meanwhile, we can test which microRNA are alternated by oxidative stress. We have specific candidates. We can use qPCR to test if these microRNA are changed, or we can use the microRNA to get a global profile change of all microRNAs. And once we identify which microRNAs are misregulated,
we can kind of correct the misregulation of microRNA. If it's upregulated, we can use anti-mir or CRISPR-Cas9 technology to delete this microRNA. If it's downregulated, we can overexpress to rescue this microRNA. And after that, we can compare if the reverse of the microRNA dysregulation could reverse the tumorigenesis effect after oxidative stress.
So although previous studies have researched the role of inflammation and mRNA in ovarian cancer pathogenesis, they have explored it separately, and there has minimal studies that's been conducted on looking at the role of fallopian tube inflammation and its effects on nearby mRNA through the generation of reactive oxygen species. So our proposal investigates these areas of limited studies and link them together to cohesively come up with a mechanism for ovarian cancer origin as well as pathogenesis. And why is this important to understand how ovarian cancer arises? Well, over the last 50 years, the survival rate of patients with ovarian cancer has not changed dramatically. And this is partially due to the fact that current management is directed towards the established cancer itself rather than the mechanisms by which ovarian cancer arises. And so understanding this mechanism will in fact lead to important implications.
And these implications can be fairly simple. We proposed that it could be possible to have some preventative measures for ovarian cancer with anti-inflammatories. Another thing that that our proposal gives is a potential target for existing ovarian cancer treatment, that being the mRNAs. And at this time, we'd like to thank you for listening, and I'd love to take some questions.
We have some, there we go.
So this is really interesting, hypothesis. Thanks for presenting. I'm wondering about, so like if you have these reactive oxygen species and whatnot creating damage, which is something that a lot of people have talked about, you would expect that to, in the early carcinogenic environment, be activating like macrophages and producing a T-cell response that would potentially attach to these like antigens or whatever and like take down the tumor before it develops. So what I'm wondering is, in your guys' model, why isn't there a T-cell response early on taking care of it? And would giving people anti-inflammatories potentially suppress a T-cell response from happening when you want one to happen early on? And third, sorry for so many questions, did you guys look at any miRNAs that might interact with the adaptive immune response?
Yeah, I think the focus of our project is on the tumorigenesis of the fallopian tube cells rather than the immune response. I think if we focus on immune response, that would be a quite different hypothesis. So I think our hypothesis really focus on how could the dysregulation of microRNA by a reactive oxidative species could change the fallopian cell themselves rather than how do they change the interaction with the immune system. So I think that's a really interesting question we can explore further, but that's out of the scope of our hypothesis.
Also, to answer your T-cell question, by the time a tumor forms, it's in sort of a hypoxic condition, which means the cancer cell is secreting a lot of lactate. And the way that the T-cell operates is that it operates in the same way where glycolysis is its primary energy generation method. And in order for it to function, it needs to expel out all the lactate that it's generating. But since the environment in which the tumor is in a lactic environment, they can't actually expel the lactate, which means the T cells die of asphyxiation.
Okay, so the genetic question again. Is there any high rate of alterations in those RNAs, in those microRNAs genes, that you could propose that not only is inflammation that is upregulating their expression, but there's some gain of function mutations of some of this have been being reported by the big studies like Bill et al. Or I don't know. Are there any one microRNA that is highly mutated?
Actually, back to the microRNAs we proposed earlier, we actually have the previous research have demonstrated that those microRNAs are either upregulated or downregulated in the ovarian cancer studies.
this is not based on the patient study, of course. This is like mostly on like the mechanism study. So since microRNA is relatively new field, it's kind of hard to quantify on like patient-to-patient base. It's not like have an expression product that you can test. It's like in the non-coding region. So like it's kind of hard to know the expression of microRNA via like sequencing or like a microarray or like some kind of like the global like patient study. So most of the study right now are focused on specific mechanisms rather than like patient-based association studies.
Other questions? Cash.
You mentioned 4 major microRNAs and associated pathways. Have you considered putting them in a sequence of events in terms of the carcinogenic changes that leads to transformation, then migration to the ovary, and then lodging in the ovary and survival there?
Yeah, I think that is a really good question. Actually, the 4 microarrays we propose, like 2 of them, they are like lead to the loss of tumor suppressor genes, which could explain how does normal cells transform into the cancer cells. And another microRNA, the kind of the downregulation of this microRNA could lead to the upregulation of the cell mobility and the cell apoptosis ability. This could explain how does like the cells with like the mutation could like have like a better migration ability to move into the ovary. So I think, yeah, this is a really good question. I think like once we got like a more detailed microRNA profile, we can have like a better story on how to explain the sequence of events.
Any other questions? One more. Let's go way over in the corner, and then we'll let you go too. We have time for 2 questions. Go ahead. Yeah.
Okay. So ovarian cancer is found in majority postmenopausal women, and my question is that does inflammation be constantly there for the upregulation of mRNAs?
I think the model we are proposing is it's more like on the chronic inflammatory settings, like which is not necessarily on the specifically, yeah, just like it's like long existing inflammation. So I think that might explain like how does like the post-menopausal female get ovarian cancer, because this inflammation really takes long time to accumulate the mutations of the microRNA and therefore lead to the transformation.
People tend to have a lot of inflammation, so there's a lot of activation, and that's actually what a lot of we left our presentation pretty open because we think that there's actually probably some link between a lot of these chronic inflammatory diseases and the development of cancers. But we don't have research to back that up yet, so that's why we left it so open.
Last question.
My question was about whether or not you think that it's the fallopian tube that's the cause of the ovarian cancer because of differences in oxidative stress that hits the fallopian tube versus, or if it's that fallopian tubes inherently have a different microRNA profile that makes them more susceptible to this sort of process.
That is a really good question. I think like this question could be explained like in either way. I think the like both of the points you raise are like possible. Because like the microRNA profile, they are different in different cell groups, so it might be possible that like the microRNAs like are like more damaging. The changes in microRNA are more damaging in the fallopian cell tubes, or that's because the fallopian tube they are like more susceptible for like long-term or like intense inflammation. I think both we are possible.
And a lot of the microRNA research has been kind of late in the last 2 years, specifically looking at the interplay with ROS. So it's entirely possible that if we look at different tissues, we can see some variation in the microRNA expression profile as well. And that could, of course, have differential effects depending on what was present, which, of course, would be a possible target to further investigate with this proposal.
Thank you very much.
Well, we are Team Bethulus. We're a team of brothers from the College of St. Benedict, St. John's University, and we have been working to peg down ovarian cancer. Hi, I'm James Bethulus.
I'm Nick Bethulus.
I'm Joe Bethulus.
And I'm Chris Bethulus.
And we have had, there'll be a quiz after. Yeah. We've been under the wonderful direction of Dr. Catherine Furness of the Biology Department at CSBSJU. So just like everyone in this audience, we were tasked with the same question. And that question was, in simpler terms, what makes good cells in the fimbria go bad?
And to answer this question, we propose that progenitor cells called PEG cells located in the distal part of the fallopian tube in the fimbriae are exposed to estrogen metabolites which induce DNA damage and cause the transformation into ovarian cancer. And along with this, our research, we focused on the main aspects found in ovarian cancer literature. The first aspect that we focused on is that ovarian cancer originates in the fimbria and not the ovary. The second aspect is that during ovulation, follicular fluid is released, and this follicular fluid bathes the fimbria, and then it comes in direct contact. And this follicular fluid contains many things, including cancer-causing molecules like reactive oxygen species, like many others have talked about, but it also includes interleukins and cytokines. And the third aspect that we try to focus on is the fact that ovarian cancer is highly chemoresistant.
The fact that this type of cancer is so chemoresistant is very interesting. To better understand this, we looked at other types of cancers that were also chemoresistant. Interestingly, certain lineages of breast cancer have stem-like progenitor cells that initiate and maintain breast cancer tumors. One such population in the fimbria are called PEG cells. So traditionally, there are two types of cells that are generally known and well known in the oviduct, and those are ciliated and serous. However, on the board here, there is also a third type of cell called the PEG cell. PEG cells express CD44, which is a hallmark receptor of stem-like cells.
So now that we have established that PEG cells have stem-like characteristics, one of these characteristics is that PEG cells can differentiate into other fallopian tube epithelium, such as secretory cells and ciliate cells. And the way we identify these certain cells is through, they each express their own proteins. So secretory cells express PAX8, PEG cells express CD44, and ciliate cells express TUB4. And now, interestingly enough, PAX8 and CD44 are expressed in and around high-grade serous carcinomas.
So like Joe was saying, this is a diagram of a high-grade serous carcinoma. And so in high-grade serous carcinomas, as you can see in the top right image, they express the protein PAX8 at high levels. Now this is interesting because we also know that the differentiated serous progeny of PEG cells also express PAX8. Furthermore, which really wouldn't be clear to anyone but a histologist, as you can see, what this image is showing in the left is that at the periphery of the center of these high-grade serous carcinomas in the distal fimbriae, there are there's a high expression of CD44, which Joe mentioned is kind of that hallmark receptor of PEG cells. So these two things together suggest an interesting mechanism for the initiation and maintenance of a high-grade serous carcinoma that develops into ovarian cancer. So it starts with a PEG cell that produces normal progeny. At one point it receives a mutation. After being mutated, the mutated PEG cell is stuck producing mutated differentiated serous progeny. These cells are the cells that comprise the body and really the main body of the high-grade serous carcinoma, and the PEG cell stays in the periphery. However, it's important to note that while treatment that we currently have is aimed at reducing that tumor mass, it is not effective at treating the peripheral progenitor population on the border. And so the question that remains though is, what is the actual cause of this cancer? What causes the PEG cell to become mutated?
So like many groups have already said, that during ovulation, the ovum is released into the fallopian tube, but also follicular fluid and cytokines. And we believe that it is these cytokines, specifically interleukins, that play a vital role in the initiation of this deadly carcinogenesis.
So this is our cancer mechanism, and I'm going to walk through it with you. So step one, you can see, like Joe was just talking about, we have an interleukin that's binding on the exterior of a PEG cell in the fimbria. Now, after binding to the interleukin receptor, this causes a pathway activation that causes an increase in the enzyme STS activity. STS is an enzyme that's responsible for converting biologically inactive estrogen into its active form. Because STS activity is increased due to the presence of interleukins in copious amounts, we have an accumulation of estrogen on the interior of the cell. Now there are two things that this accumulation of estrogen at a high density causes. First, we know that estrogen at high levels in cells causes superstructural changes to chromosomes and chromatin. And well, this is incredibly significant because what this means is that in PEG cells, at every single, well, it could be a reason that at every single ovulation, the DNA is being relaxed and decondensed into a state where it's vulnerable to attack by mutagens. Now, additionally, at the same time that we have those DNA superstructural changes, you also have the enzyme CYP1B1 metabolizing estrogen. And at high levels, the metabolites of estrogen build up, and some of these metabolites we know cause cancer. One of these is 4-HE. And together, so we have DNA that's exposed and an accumulation of mutagens. This is kind of the perfect storm where you have regions of the DNA that are exposed and that are being assaulted by these estrogen metabolites. Together, this leads to repetitive and cumulative damage to the DNA that over time results in the cancerous PEG cell phenotype and genotype.
So in summary, a PEG cell undergoes a mutation described in the process that James just explained, and then its progeny multiply and form what we call high-grade serous carcinoma. And now, due to its proximity to the ovary, this high-grade serous carcinoma spreads and resides to the ovary and forms what we know as ovarian cancer.
So to revisit our hypothesis again, we propose that progenitor cells called PEG cells located in the fimbria are exposed to these estrogen metabolites which induce DNA damage and cause the transformation of epithelial cells into ovarian carcinoma.
So for the significance and innovation of our hypothesis is that we have identified a novel progenitor population in the fimbria. This, if our hypothesis is correct, this will lead to a paradigm shift in the ideology of how we view ovarian cancer, which will lead to hopefully more improved treatment options.
And so we propose initially doing three things. One, it's important to note that this specific type of cell, like Nick said earlier, has been largely overlooked by researchers. I would challenge you to go out and try and find a definition of what a PEG cell is, because we couldn't, it's not clear. And there's only been one paper published to date that has correctly characterized this cell type. So we really need to do some basic assessment, and that includes flow cytometry to assess what's on the outside of this PEG cell, and that would allow us to develop new treatment types that I'll talk about in a little bit. Another valuable tool, and there's also numerous grants for this, would be single-cell monitoring. And single-cell monitoring would allow us to better characterize the microenvironment of this cell, which would, again, help us make better informed treatment programs. Finally, we don't know what protein is being expressed in this cell type, nor do we know what level it's being expressed at. Therefore, we propose using RNA-seq to determine both of those things, so we can kind of paint a picture of what a PEG cell is, and hopefully that will help further the understanding of our hypothesis. Now, interestingly, progenitor cells, so the way that chemotherapy works is you deliver chemotherapy to a cell, and it causes the cell to lyse. That's one of the ways that it can work, by causing so much damage. And interestingly, progenitor cells and stem-like cells are resistant to this lysing or apoptosis. And there are a few different reasons for it, but no one's 100% sure why. However, in a certain form of breast cancer tissue, in this cancer line, there is a progenitor population that, like we're proposing in the ovary, initiates and sustains a tumor's population. And one proposed treatment that is being used currently in mice is the delivery of chemotherapeutic agents with nanoparticle drug delivery using a polymer-coated vesicle. Now, what's really interesting is that progenitors that are cancerous, like our PEG progenitor, we hope, express isoforms of CD44 that are unique and unlike the rest of that don't take the same shape as the rest of the CD44 receptors that are on healthy, normal stem cells. Now, because these cells, because the CD44 on these cancer progenitors are different, we can exploit this, and researchers have exploited this to target vesicles, and essentially these vesicles will be taken into the cell through these receptors. This increases the concentration of chemotherapeutic agents in the cell to a level that would actually cause it to lyse. So therefore, we've kind of met that problem with having chemotherapy at too low of a dose inside the PEG progenitor, and if we can ramp that up, it will cause the cell to go under apoptosis, or essentially burst. So we've said a lot of things, and we thank you for sitting so patiently, and now we'd like to invite any questions you may have.
Questions from the audience? Go right ahead.
First of all, I want to congratulate you for your own figures. You guys made more your own figures.
Could you tell?
And I think that was terrific. I mean,
thank you.
Do you have any idea? So this idea of the stem cell, cancer stem cell, is developing rapidly.
Yes.
Do you have any idea how this population might have self-renewal and at the same time give rise to secretory and ciliated cells? Let me just finish for a second. Normally cancers are highly undifferentiated, right? So PEG cell could be the origin. The question is, does it differentiate into these two other cell types that we see the most in serous great carcinomas? Do you require less hits to be able to differentiate? And then do you have any ideas of how this may happen?
So let me make sure I got your question right. So what you're asking me is, why is it that it just produces serous progeny in the tumorous form?
The question is if it's really the source of origin of the tumor, right? If it has enough genetic mutations, right, to become the tumor, how is it that it can give rise to specialized cells like secretory and ciliated?
Yeah, that's a good point. Well, actually, what we found is that after it becomes, after it has that tumor status, it has locked into a certain progeny that we see as in high-grade serous carcinoma. And to me, what that would suggest, although we did not stipulate, we wanted to, again, be very open-ended in this, but what that would suggest to me is that there could be something going involved with like a fusion gene or a faulty repair mechanism that causes the hyperexpression of proteins that would promote a certain lineage. So that would be one explanation, but I think that's a good question, and I think, furthermore, we just don't know enough about PEG cells, and I really couldn't give you an informed answer because of that.
Go ahead.
So, do you know specifically are PEG cells only found in the ovary and the fallopian tubes, or are they found all over the body?
Good question. So, do you want to take this on it?
Okay, so they are only found in the fallopian tube, and they are concentrated specifically in the distal end, which is really important, since mostly this type of cancer is in the distal end of the fallopian tube.
Yeah, and so what? And additionally, are you going to say something?
No, I'm good.
Okay. So your question though is like, so what is? I guess that really gets to the bigger question of what is a PEG cell? Well, a PEG cell is literally a term used in histology to describe a progenitor that. So this theme is wide throughout the human body. In your skin, you have progenitors like this. Even in your brain, but so yes, the progenitor in this region of the body is called the PEG cell.
Okay, so my question about that is, can estrogen affect other progenitor cells around the body? And if so, do people who take estrogen supplements have increased risk of cancer throughout their body?
1 second. Okay. So yeah, that's a good question. So it's very interesting that you would bring this up. So the estrogen hypothesis, a prerequisite to having this cancer is having the cellular machinery to metabolize estrogen and even to take it in. So, well, I guess estrogen can go in any cell, but to really metabolize it and have those receptors. So, this is seen in breast cancer, which was the basis for why other researchers have come up with an idea to kind of exploit that feature. But additionally, we know that when you reduce the levels of estrogen in some as a treatment for breast cancer, the breast cancer kind of is not eliminated in a sense, but it does kind of tone down.
We're going to have to call time here. We'll have more time to talk with you, your poster, because clearly you've generated a lot of questions. So, well done.
Hi everyone, we're the team from Carleton College. My name is Shraddha.
I'm Rashida.
And I'm Preethia.
And our presentation today is about the activation of AKT as an initiating event in high-grade serous carcinoma. So this is the roadmap for our presentation. We'll start with the background and introduce our hypothesis, and then describe the connection between ovulation and HCSC. Then talk about the activation of AKT in tumorigenesis, and finally end with regenerative and therapeutic techniques. So epithelial ovarian carcinoma is a lethal gynecologic malignancy, and about 60 to 80% of these are characterized as the HCSC histological subtype. 85 to 90% of these cases are due to sporadic mutations, and recent evidence based on salpingo oophorectomy samples suggests that HCSC may arise from the epithelium of the fallopian tube. Specifically, these studies have found a precursor lesion, serous tubal intraepithelial carcinoma, also known as STIX. So from this background evidence, we narrowed down the original question to primarily focus on neoplastic transformation that leads to serous tubal intraepithelial carcinoma, or STIX. And we hypothesized that the formation of STIX are induced by the activation of AKT, which occurs due to changes in the microenvironment of the fallopian tube epithelium during ovulation. And specifically, these microenvironmental changes are due to inflammatory response following ovulation and retrograde menstruation. So there have been several epidemiological studies that have suggested a negative correlation between anovulation and the rate of acquiring ovarian cancer. Particularly, anovulation has been linked to the use of oral contraceptives, parietal, and breastfeeding habits. Now we'll take a closer look at the process of ovulation. Here we see in this figure the inside of an ovarian follicle. The oocyte and the adherent cumulus granulosa cells surrounding it will be released into the fallopian tube, and this results in the exposure of the distal fimbria to the surrounding environment containing follicular fluid. Due to this, the microenvironment is rich in inflammatory cytokines, and over the reproductive span increases the susceptibility to tumorigenesis.
So our hypothesis is based on the activation of AKT as an initiating event in tumorigenesis. And AKT is an oncogene that encodes for a kinase that is activated by several pathways implicated in cancer, apoptosis, proliferation, migration, DNA damage recognition, and repair. In the case of high-grade serous carcinoma, activated AKT has been found in the fallopian tube secretory epithelial cells, and several mouse models have shown that aberrant expression of AKT is required in tumor formation. This diagram here illustrates the AKT pathway, showing that P13K phosphorylates AKT, localizing it to the cell membrane and causing downstream effects. So together, this P13K AKT mTOR pathway is really crucial in regulating the cell cycle. So this makes sense that the activation of AKT initiates tumorigenesis because it disrupts DNA repair mechanisms and suppresses apoptosis. So this is our working model that we'll be stepping you through as we explain our hypothesized mechanism. We're focused in on the microenvironment surrounding the distal fallopian tube. And at a basic level, we see that both inflammatory responses and retrograde menstruation play a role in STIC formation through activation of AKT. So the first factor that we propose leads to the activation of AKT is oxidative stress elicited by inflammatory cytokines in following ovulation. And oxidative stress has been shown to affect the epithelium and the underlying stroma in the fallopian tube. And a recent study has shown that production of hydrogen peroxide during inflammation directly upregulates phosphorylated AKT in three-dimensional organ cultures. And these results are shown. So if we focus in on the top row showing the green staining is phosphorylated AKT. So at a basal level, we don't see the phosphorylated AKT. But when 1 millimolar of hydrogen peroxide is added to these organoid cultures, we see that phosphorylated AKT is robust. And the same results are shown in the Western blot. In the H column, which is the hydrogen peroxide treated organ culture, we see an increase in phosphorylated AKT when compared to the B column or the basal. So when we add this into our model, we see that inflammatory responses in the follicular fluid contains TNF-alpha, and TNF-alpha interacts with
with reactive oxidant oxygen species, and this elicits an oxidative response, an oxidative stress, which activates AKT. Another component of this inflammatory response that we hypothesize activates AKT is fatty acid synthase. And the follicular fluid is not only rich in inflammatory cytokines, it also contains steroids, and the overproduction of steroid hormones is linked to the regulation of FASN biosynthesis and expression. And a 2010 study analyzing sticks showed that FASN overexpression was significantly found in these sticks. So we linked that study to a recent study showing that there's overexpression of fatty acid synthase in epithelial ovarian carcinoma, leading to the activation of AKT. So in our model, we also included this in the follicular fluid, steroids, which interacts with FASN, and then FASN goes and upregulates the activated AKT.
So in addition to oxidative stress and FASN, exposure to catalytic iron is another process that can activate AKT in distal fimbriae. Several studies have shown that during retrograde menstruation, high concentrations of heme and iron is exposed to distal fimbriae. Here's a figure from a study that looked at the ratio of RAS-positive cells in response to iron. So they found that the percent of RAS-positive cell increases with iron in a dose-dependent manner. So as you increase the concentration of iron, the ratio of RAS-positive cell increases too. RAS is a protein that has been implicated in the AKT pathway. So RAS can actually activate the AKT pathway through PI3K AKT mTOR pathway. So the authors of this paper concluded that RAS can actually activate AKT pathway, and they also quantified AKT, but they didn't have the figure, so we can't show that. In addition to that, exposure to high concentrations of iron can also result in reactive oxygen species production, and as talked before, ROS can actually again lead to AKT activation. So in our model, we included retrograde menstruation that exposes high concentrations of heme and iron group to the distal fimbriae that can again lead to AKT activation. So in order to test our hypothesis, we proposed organoid culture and experimental design. A new study by Kessler et al. Showed that using a single epithelial stem cell, we can culture fallopian tube organoid in vitro condition, and then this fallopian tube that has been cultured in vitro condition contains both the ciliated and secretory cells. Now we can treat this the organoid culture with follicular fluid that has been extracted from patients, and then we can quantify the expression of various inflammation-related genes, and we can also quantify the level of AKT activation. So for the possible therapeutics, we propose that we can use anti-inflammatory agents such as aspirin, ibuprofen, and fish oil to block the inflammatory response. Or we can also use the small molecule inhibitors of AKT to directly block the AKT protein. So with that, we have the references, and we'd also like to acknowledge Professor Debbie Walter-Cones at Carleton College, Katie Campbell, Dr. Scott Kaufman, and Dr. Larry Carnage, and the whole Mayo Clinic Impact team.
So we have time for questions. Go right ahead.
Another team already included the possibility of taking aspirin as an inflammatory reduction. Did you find out how much aspirin and how often that they would have to take?
I don't know. I think it was like an observational study, so it was just looking at the amount of NSAIDs taken. But there is, I don't know the exact statistics, but there is a correlation between taking NSAIDs and it's negatively correlated with getting high-grade serous carcinoma.
Cordelia?
With your implication of the retrograde menstruation pathway, do you see any changes in ovarian cancer risk with women with endometriosis?
I believe there was an increased risk. When we looked at epidemiological studies, there was a lot of factors that increased, with an increased risk of pelvic inflammatory disease, and endometriosis was also one of them, that if they had that, they also had an increased risk of getting high-grade serous carcinoma.
Larry?
So ovarian cancer is driven by genomic alterations, mutations, and other types of lesions. What's the link between your activation of AKT and the development of those lesions that would actually cause cancer?
So what we proposed was that this AKT activation in the fimbria cell, in the distal fimbria cell. So during ovulation, when the distal fimbria actually kind of brushes up the ovaries, some cells are actually transferred, and people have talked about the metastasis and how the cells can be transferred to ovaries. And that's our link, that the cells with these mutations can actually go to ovary and cause the ovarian cancer.
So to piggyback on Larry's question, have you found mutations in the AKT-PA3 kinase that are prevalent in ovarian cancer patients? You know, anything downstream, target genes, upstream, right?
So there have been studies that have actually looked at the level of AKT expression in high-grade serous carcinoma, and they have found that AKT is overexpressed in high-grade serous carcinoma.
Other questions? Okay, I think I'll thank you very much.
I'd like to thank everyone for their attention. We're the last ones presenting right before lunch, so hopefully we can hold your attention here. My name is Rebecca, and this is Quinn. We're representing Anoka Ramsey Community College, and we're going to be discussing the topic investigating and tracking the cell of origin and genetic hits required for sporadic fallopian tube epithelium transformation to ovarian cancer. Ovarian cancer, as you've heard throughout the day, is the leading cause of death among gynecological diseases, and it afflicts over 200,000 women and claims the lives of over 125,000 women worldwide every year. The nonspecific signs and symptoms that are associated with the ovarian cancer disease cause significant delay in the diagnosis, and it often is not diagnosed until stage 3 or even 4, by which time it has metastasized. The long-held view has been that the ovary itself is the site of origin for ovarian cancer. However, the current view has been transitioning by many experts to the idea that the site of origin is actually within the fallopian tube epithelium. There has been shown a strong correlation to a large number of menstrual cycles and wound and repair actions that accompany ovulation and the menstrual cycle. The wound and repair actions occur both within the ovary itself and also at the distal end of the fallopian tube. Depicted here, and I'll see if I can, maybe, okay. So depicted here is the fallopian tube, and we saw this in another presentation. So the normal fallopian tube epithelium here, and it's depicting the secretory and ciliated cells. Peg cells are not depicted in this representation. And so with, as the ovulation occurs repeatedly throughout menstrual cycles during a woman's life, with every occurrence of the ovulation, some of the cells are exposed to these ROS and other inflammatory things. As they are continually exposed to these inflammatory proteins, they develop this P53 signature and start to develop mutations. And as they progress, they accumulate more mutations until they reach the stage of tick cells. And then beyond that, they move on to the invasive serous carcinoma stage and progress on. The number of stem cell divisions is strongly correlated to cancer risk, with greater number of stem cell divisions indicating greater risk of cancer recurrence. Endogenous mutations occurring during DNA replication have a greater impact on cancer incidence than either environmental or hereditary factors. As seen in this figure, adapted from Tomasetti and Vogelstein in their recent article just published in Science magazine, the total number of stem cell divisions and the lifetime risk of developing a cancer greatly increases as the number of stem cell divisions increases. And ovarian cancer is represented at this stage. Also within the Tomasetti and Vogelstein study, they used biostatistical analysis, which please don't expect me to try and explain, and they submitted all these different types of cancers to their biostatistical analysis and drew the following conclusions: that there is key distinction. All of the cancers indicated by the green bars are ones that are strongly associated with replicative disorders. So with every increase in replication, there is an increase in the incidence of these cancers. And the blue bars indicate diseases that are noted to have environmental or hereditary factors. And ovarian cancer falls in the middle on this chart as well. So PEG cells that we just learned about from several other groups. Given the evidence for the importance of stem cells in many cancers, PEG cells have been proposed as fallopian tube epithelial stem-like cells. The PEG cell has both clonal growth and self-renewal potential. PEG cells express epithelial cell adhesion molecule, or EpCAM, CD44, and integrin alpha-6. However, they lack the markers for ciliated or secretory cells. They are highly concentrated in the fimbriae of the fallopian tube, and they have a 5-fold growth when compared to other fallopian tube epithelial cells.
So an important factor that we need to discuss is the genes that plays in oncogenesis. The only gene that's shown in ovarian cancer that's prevalent in all ovarian cancer is P53. P53 is a tumor suppressor gene that drives apoptosis, senescence, and autophagy. The other gene that isn't well characterized is PI3 kinase. As we just learned, PI3 kinase is also AKT. PI3 kinase is a gene that drives cell proliferation and extends the life of a cell. And what we found in the literature is that PI3 kinase and P53 has a feedback loop with one another, and they drive the rate of apoptosis or proliferation. Here, we show that PI3 kinase inhibit P53 through MDM2, and P53 inhibit PI3 kinase through P10. So our hypothesis is that overexpression of constitutively active PI3 kinase and loss of TP53 activities in stem-like PEG cells are required lesions leading to transformation of fallopian tube epithelial cells to high-grade ovarian cancer. So what we're proposing is that we disrupt that two feedback loop where P53 is no longer present and the cell just continuously divide because PI3 kinase is overexpressed. Our rationale is that PEG cells are a great candidate for the cancer stem cells because their ability to maintain and replace damaged fallopian tube cells during ovulation. And the rationale that Rebecca explained, that the number of cell division in a stem cell is highly correlated to a higher risk when there's more cell divisions. So when we looked up P53 in the CBio portal database, which is our website that we could look at specific gene mutation on the website, we found that P53 has an 88% mutation in our ovarian cancer. In the green, the green shows that this mutation is actually a missense mutation, while the black shows a truncated mutation. So P53 is a common cancer that's seen that has a loss of function. When we looked at PI3 kinase, we found that PI3 kinase is also mutated, and there's a lot of overexpression where they're being amplified. And this is interesting to us because not a lot of literature we found was talking about PI3 kinase with P53. So what makes our proposal unique is that, one, we're proposing PEG cells as a cancer stem cell, and two, we're proposing to disrupt that two regulation of the two genes. And what's most unique about our proposal is that since there is no in vivo model that we could use to study cancer in mouse, we are proposing to generate a transgenic mouse and cross it with a knockout mouse. In the transgenic mouse, we are constitutively activating PI3 kinase under the EpCAM promoter, and we are crossing it with knockout P53 with a green fluorescent protein, and we're going to cross that mouse so that we could track them. So with this F1, hopefully we'll get that F1 generation mouse with a constitutively active PI3 kinase and knock out PP3 and green fluorescent protein. We are able to track the cancer from the start to late stage.
So why does this all matter? What are we trying to get across with our proposal? We're trying to present an in vivo model that has not yet been able to be represented that can truly recapitulate type 2 ovarian cancer, where the cell of tumor origin can be tracked and combination therapy involving PI3 kinase and a peptide inhibitor can be tested, and will provide new treatment options for these cancer patients and decrease the number of deaths associated with this disease. A recent article that just came out in, I believe, January attacked a totally different problem, but with a similar presentation, and where they worked with zebrafish and used a single cell expressing an early neural crest marker and tracked it through transformation into melanoma. So I'd like to take a moment to thank everyone here at Mayo for the opportunity to come in down and present, and to Regenerative Medicine Minnesota for sponsoring this, to Dr. Starr and his lab at the University of Minnesota for exchanging ideas and giving us the opportunity to practice and discuss things, and a special thank you to our advisor, Dr. Paula Kronquist.
So we have a couple minutes for questions, one right there.
What would you suggest causes the upper regulation of the PI3 kinase?
Okay, so we're suggesting, as Rebecca explained, that cancer cell, in ovarian cancer cell, the rate of division increases. If you have a high rate of division, there's a high increase of cancer risk. So we're proposing that this is mostly endogenous error where it's replication error that creates this mutation.
Other questions? Go right ahead.
I see the data like up here is about just ovarian germ cells, so are you just extrapolating to the fallopian epithelium from that?
One question over there, go ahead. No, go ahead. Got the one sitting on the aisle first, okay?
So in your proposed testing method, you said you wanted to knock out P53.
Yes.
P53 is expressed in basically all of the cell types, so I was wondering how you would propose cutting out P53 from all of the cell types, and if you do manage to succeed, if that phenotype is lethal to the potential fetus that you're treating.
Right. So what we're proposing is that P53 knockout is under the OVG1 promoter, and that's specifically to the Mullin tract, which develops into the fallopian tube in the uterus. So we're only expressing knockout in those cells.
One last question.
Yeah, I think kind of question build up on this question is like, it seems like the OVIGT1, the promoter, is like a ovarian specific promoter. Like it will like knock down like P53 in the whole like ovary, like whenever this promoter generated. But how do you like specify like fallopian tube versus the other ovary cancer, given like, imagine that it's really hard to find like a fallopian tube specific promoter.
So you're asking like, how do I tell the difference between like a PEG cells versus like a secretory
No,
cells?
I'm thinking about like this promoter actually is a promoter for the whole ovary rather than fallopian tube specific. So I'm wondering like, how do you differentiate whether the cancer type generated from the ovary or the fallopian tube?
So we can look at markers of PEG cells, and they express EpCAM and CD44 and integrin alpha 6. So we can use those markers to specifically track those cells.
I think like I'm more wondering about the origin of cells. It's like you are focusing on the fallopian tube. You want the cell origin to be on the fallopian tube rather than the ovary. And it seems like the promoter drives both the fallopian tube and the ovary. Like, how do you differentiate both? Thank you.
So I'm going to jump in at this point and suggest that we continue that discussion in the poster because, in fact, I'm well aware that for the last 15 years, folks have been trying to find a fallopian tube-specific promoter so they can do these sorts of studies. So I think it's a little unfair to burden this team with that. Okay, we have about 2 minutes left, and so I'd like to make a number of points. First of all, for all of you who presented, congratulations. This was your first presentation for many of you in a scientific forum, and well done. So, a couple of other points. Hopefully you're getting the sense that participating in science and talking about science is a lot of fun, because that's what drives those of us who are in the room who are scientists. Coming up with ideas and testing them. The other idea that I think you should take away is that given a certain body of literature, like there is on ovarian cancer pathogenesis, you can come up with lots and lots and lots of different hypotheses. And it's not a question of right or wrong, it's a question of simply going into the laboratory, proposing an experiment, testing it, discarding those that don't seem to fit, and continuing to move forward. And that's what all of us do every day. So congratulations on coming up with your hypotheses and your means of testing them. And then the last point I want to make is that all of us have mentors. In fact, I was on the phone yesterday with my clinical mentor who taught me everything I know about the disease that I take care of clinically. And so all of us have mentors. We rely on them every day. So I'd like the mentors for these teams to stand up for a minute so we can acknowledge them.